Okay, should we get started? Start sharing screen. Wow, I'll have to zoom out, I guess. How do I zoom out from here? Wow, so much blank space in these figures. All right. Is Amanda doing figure 1?
Yeah.
Okay.
Okay. So figure 1A is basically they wanted to see how the mixture of sweet and bitter tastes interact and how the mice react to that. So judging, they didn't say which was which, but judging from the graph, the Ace K is probably the sweet, and then the gray is probably the bitter, and then the red is the combination of the two. And you can see that they didn't lick as much when there was a combination of sweet and bitter, and that was pretty much all they said about that.
Yeah.
For part B through D, they wanted to see the projections in the brain, and so they labeled neurons in the bitter cortical field, which is the GCBT neurons right there.
So wait, Amanda, so what happens when you combine sweet and bitter?
They have like an adverse reaction to it, and they treat it just like the plain bitter stimulus, which means they don't interact so well.
Right.
So the bitter, I guess, just overpowers the sweet.
Yeah. So Jahao, this is like what we were talking about, because obviously this project didn't start off with this question of whether bitter suppresses sweet, right? But this one figure 1A was added on like probably at like the last minute, just to exemplify what they're trying to talk about. And therefore this story becomes more about like how does bitter and sweet interact, rather than a paper on finding different markers for the NST, the NTS. Right? So this kind of a simple experiment to show what the story really is about, I think, is powerful, even though it's like a very simple, obvious figure. Right? And same for us. If we taste something bitter in anything we eat, we're going to spit it out, no matter how good that dish is. Yeah. All right. So then, after that.
Yeah, they were saying that bitter was important in the introduction for toxicity, like potential. It's like an evolutionary thing, and then sugar's more for energy. So maybe that's probably why the bitter overpowers the sweet, is because it's like a body defense mechanism. But for B through D, so the GCBT neurons are in the bitter cortical region, and then they also looked at neurons in the central amygdala. Tried to find more information about that, couldn't really find much online about why they picked the central amygdala. But then they said that both areas are key to encoding the identity of the taste stimulant. And then they labeled them with TD tomato and GFP different colors, and then looked at the projection to the NTS, specifically the rostral NTS. And they said that the rostral NTS is the first sign of like the signals from the periphery into the brain, and that it traveled all the way from those areas in the brain to the brainstem. And then they concluded that these long range projections suggest a top-down control of taste behaviors. And I know what top-down is, and I know what they were showing here, but I can't understand why it shows a top-down processing. So that's kind of where I was getting stuck. But that's basically the entire figure, is that both of them project all the way down to the brainstem and the rostral entities. So that was it. They just didn't explain some things. I had to figure some things out on my own, but they just threw in that conclusion sentence, but I couldn't figure out why they think it's top down just because they project to the brainstem. I don't know. So that was my question.
So yeah, so here they're looking at projections coming from the cortex or the central amygdala into the brainstem, right? And they're just assuming that because there are projections coming from these areas, that synapse onto the neurons in the brainstem, that they'll be getting some feedback information from the cortex, right? Because the way that we think about taste sensation is that it goes from the tongue, ganglion, brainstem, and then up further into the brain, right? And that it's an ascending pathway. But here, just from the anatomy, they're suggesting that there's some information that might come back from the cortex that will modulate, regulate the information that gets passed, as that it's like a feedback loop, that some of the information that goes up to the brain is already kind of regulated within the brainstem. Yeah. Just purely on anatomical structural results. Does that make sense? So it's a pretty simple figure. Right? So should we move on? Okay. So in figure 2, I mean, I've been talking about these results for a while now, so I hope that most of the data, if not, I mean, we haven't seen the data, but I hope that they are quite obvious in what they're trying to show. So who's going to explain figure 2 to us?
So they are testing how the different, how the neurons in the rostral NTS respond to different taste. So they first want to make sure that all the neurons will respond to all 5 different taste qualities. So that's why they inject the sync GCamp, try to monitor the response of the neurons by fluorescence. And they see that compared to water, all the 5 major taste quality will have a response in these neurons. So, and then they look at specific markers that has been previously established, for example, somatostatin, SST. So they use SST, they inject them into the SST mice, and they see that only the bitter tastants were activated, which means that SST neurons are specific to bitter. Similarly, they find that COP2 is specific to sweet, both to sugar and to artificial sweetener. Yeah, so basically they're testing previous established neurons.
And how do they find these markers?
I think it's similar to buprenorphine, right? That is also from the Allen Brain Atlas. They try to find a marker and then they test it and find it happened to be the same. Right. So I don't know. I'm curious, when they are trying to find a marker, do they just select from the top or do they have an educated hypothesis saying certain marker might be more candidate than the other?
No, just buying a lot of. I think the pipeline was first finding on Allen which genes are expressed in the NTS.
Mm-hmm.
Second cutoff is are these mice easily obtainable? So are they sold in Jackson? And then the ones that they could get, they just did photometry on.
So for a project like this, for example, before they find a buprenorphine to be specific to sour, like how many other have they tested? Or they just like, this is the first one they tested?
Maybe in total about 20 strains.
Oh, wow, okay. I see. So it's a large work.
Just a question here. Do you know what this means? This like double colon?
Mm.
Does anyone know what this means? Any yeast biologists here? No, nobody's seen it before. No. This just means that this gene is driven by this promoter. So it's a common genetics way of denoting promoter and insert relationship. So although this is subtle, here you can see that they're injecting synapsin GCAMP that's driven by the synapsin promoter, as opposed to SST hyphen GCAMP, which probably means that they injected GCAMP, a Cree-dependent DIO GCAMP into an SST Cree animal. And the
So
same
I think this is where I was missing a lot of the information. It's like, what happened? What did you inject? What's the experiment?
Yeah. So the first pass, what they did is they injected synapsin GCAMP, which will into the NTS, right? We've all done that, injecting into the NTS, right? Here they're injecting the GCAMP reporter into the NTS and then hooking up with phytophotometry, different taste qualities on the tongue. And I think these were not hungry, thirsty animals, and they're trained to lick with a cue.
Didn't they do it with a cannula?
No, I think I don't know if these figures were with the cannula, just because of that panel A. They did do some things with the cannula.
Yes, but it doesn't show in any of the figures, so I'm just presuming they might have done it with a cannula, but not show it because it's not a very nice figure to show.
Yeah, that could be also. What do they say here in the figure legends? Oh, it is intraorally.
Yeah.
So they present different tastings through the cannula and look at responses. And because GCAMP is labeling or expressed in all neurons in the NTS, then you can see that NTS neurons respond to water, sweet, bitter, sour, salty, and umami. Yeah. And this is measuring activity in bulk, right? In all neurons in this region. So then their goal is to figure out if there are subsets of neurons that convey just the bitter information, just the sour information, sweet, salty, umami, and so forth. Here they're talking about bitter and the sweet populations labeled by SST and CalP2. Yeah? Does that make sense?
Mm.
I have a question.
Yeah.
They can using a P2X3.
I missed your first part.
Maybe they can using a P2X3
Yeah.
for the taste, but
Mhm.
why they using all the neuron? Why they target all the neuron?
Oh, so if they were to use P2X2 Cree and then put GCamp, the GCamp will be in the ganglion, but not the NTS, right? Yeah. So here they want to look at activity in the NTS. But if we were to do that experiment with P2X2 and GCamp, we probably would see taste-dependent responses because there will be GCamp in the terminals. So the fibers that project there would light up according to different tastings that are presented on the tongue. So I would expect us to see something similar to this, but not coming from the cell body, so it'd be coming from the projections. Yeah. Okay. Can we move on? Yes. All right. So what happens in figure 3? What did they do?
So they basically ablated the specific neurons using a Flex DTA, which is a diphtheria toxin. So into the, I think it was Cree mice, they injected the diphtheria toxin and they looked at licking of, well, quinine. So before they didn't lick the bitter taste, and afterwards they did. I don't remember. There's not a lot to say about this. They also do it, so they did it with the SST Cree mice. They showed that for bitter there is a change, and then for sweet there isn't any change. And then with the other mice, they showed that for sweet there is a change, and for bitter, or no, for salty there isn't a change. And then,
So
I'm sorry, there's a lot of noise on my end.
so they're killing these cells, right?
Yes.
They're killing here. They're killing the SST. They don't really explain this well, but I would assume that here they're killing the SST neurons.
Yeah. Exactly.
It's not in the legends, so.
No, it's all an assumption.
So let's say they kill the SST neurons, and see whether the animals will now lick bitter substances, and they seem to do that.
Yes.
So then when you put this into bar graphs, you see that by killing the SST neurons, you're making them drink as much quinine as water, right? Because this is the one-on-one
Water, yeah.
ratio, 1-to-1 ratio. And even at higher concentrations of quinine, they're drinking this quite significantly. No changes in water, no changes in sour. So they're still rejecting
Sweet.
sour. Oh, yes.
So no change in sweet, no change
So
in
they're,
sour.
they're still drinking more sweet, 4 times as much sweet than water,
Mhm.
and about half as much as sour as water. Right? So this probably means that SST neurons are conveying bitter information, but not sweet or sour.
Mhm.
And then they do this with CALB2, the sweet neurons. So they kill the sweet neurons. And then the preference for sweet goes down from like 4 times as much to about twice as much. And even at higher concentrations, same thing. No changes in salty, no changes in sour, bitter
Other tastes.
or sour.
So they show it's specific.
Mhm.
And then
I think we can move on. Oh, these figures are so like spread apart and big, a lot of blank spaces.
this is why I said it feels like it's not ready to be published.
But all the information's there.
Not all. Some.
Figure 4.
So for figure 4, they moved on to using optogenetics to do like the opposite of figure 3 and to activate the bitter neurons and the sweet neurons specifically using optogenetics. So that's the schematic in figure A.
Mhm.
And then in B, it shows that every time the SST neurons were activated, there was a significant decrease in licking. So the SST neurons suppress the licking behavior, both B and C. And for the opposite, in figures D and E, activating the sweet neurons, the CALB2 would lead to an increase in licking behavior. You can see D and E. And then after that, they did a recognition test using a 3-port system, where in the middle port, it would randomly secrete a tastant, and the mice were trained to go to either the left if it was bitter, I believe, and right if it was sweet.
Mhm.
And so for the panel G, it sees that when it was with a high accuracy, the mice were able to go to the right tastant spouts. And for those when it was just secreted with water and a little bit of salt, that was also activated by the sweet neurons. It was the mice were trained to go recognize it as a sweet. And when the SST neurons were activated, the mice were trained to recognize it as bitter. So they went to their corresponding spouts.
Mhm.
And then finally, for last panels here, they did a four-port taste recognition test, where on one side it was secret, or it randomly secreted bitter, sweet, and salty. And on the other side of the box, there were 3 spouts that corresponded to sweet, salty, and bitter. And the mice were also trained to go to the appropriate tastant. And in the novel section, they actually secreted a fourth tasting that was more novel. And since the mice weren't really trained for that during the training phase, it shows that there was a random, they randomly selected the 3 spouts, and when the bitter neurons were activated without the genetics, it all went to the corresponding bitter spout.
I think one interesting thing is when they started doing this 3-port assay, then that they added this salty condition. And this was because so you would think that you do sweet, bitter, and then you present water, right? And that gave it more of a left-right randomness. So there was no preference for which side the mouse was going to go. So then to correct that, they started training the animals for salty to go to one side to see if they could reverse that by switching light on. Because if something's sweet, then it will override that mild saltiness. Or if something's bitter, then it'll override that mild saltiness, and bitter would win out. So this adding this sodium chloride condition was a significant improvement.
I have a question. So
Yeah.
does that mean in the training for figure G, sodium chloride was present at the same size as quinine, while in figure H, sodium chloride presents at the same size as the ACK, right?
Oh no, so what is happening here is in the middle port is where all these solutions are presented. Yeah, so let's say in figure G, this CalB2 animal, CalB2 Cree, injected with chanorodopsin in the NTS with the fiber sticking out, right? They go to the middle port and then they taste it. These are thirsty animals, very motivated. They taste whatever comes out. And if they taste, let's say ACE-K, sweet, they have to go to the right port and then they get the water reward. If they go to the left port, they get no reward, right? And then if they taste bitter, then they have to go to the left side where they will get the water reward. On the right side, you don't get any reward. Right? So left and right are presenting just water.
Okay.
Yeah, and that's the issue, because if instead of using salty, they present water in the middle, then they get confused with, like, is this the reward? So that's why they started putting in the salty, so that they can differentiate that from the left-right reward, water reward. Yeah. And John, what the list of things that you got and the diagram is to build the first step of this 3-port box. It's the 1-port box where essentially just presenting different things and measuring the number of licks. Right. At least we're moving really fast.
Oh, I have another question for the pulse figure. For the, I think B and D. Yeah. Oh no, yeah, B and D. Does the 30 hertz, or not 30 hertz, like 30 nicks in 5 seconds, the maximum that the mice can do?
I think like 7 licks per second.
So it's the maximum.
It's kind of the maximum, yeah.
Okay.
Yeah.
That makes sense then. Because you have basically, you know, like without any simulation in B, you have the maximum nick while in D, upon the activation of the switch, you also have the maximum NIC. So presumably the basal condition of the my animal should be different in these two tests.
That's right. Yeah, and there's a ceiling effect because they can't go faster than 30. Right? Okay, what figure are we on? This is 5, right? And then what happens here? So here they start introducing the combinations and doing photophotometry. So if they're measuring CalB2 sweet neurons, then they see a response. But when they present a response to sweet, if they present sweet plus bitter, they see a, I guess it's not significant. They see a slight, is this significant? It can't be significant. They see a slight reduction. Not significant, but slight reduction in the responses there. Then they look at the bitter neurons. When you combine bitter and sweet plus bitter, no change. So now they want to see if this kind of change that was observed in A comes from that projection that they had talked about in figure 1. So what happens if you activate the bitter cortex while this animal is tasting sweet, and they see now that there's a reduction of responses. So this presumably means that the tongue is detecting sweet, ganglion is detecting sweet, but by the time that it reaches the brainstem, that there's a signal coming from the cortex that says there's something bitter, so that sweet neurons have to be suppressed.
When does the night start?
When does what?
When does the CHR2 turn on?
I have no idea. Let's see. Methods. Did anyone find it? Star methods. It just says that light was delivered in a 10-second time window when taste was infused.
So presumably like at the same time when
Yeah, I would guess so.
okay.
Similar times.
Also 10 seconds. 10 seconds of night delivery. Okay.
And how long is the infusion for?
I think it's about 10. previous figure is all 10, kind of.
10 seconds?
Yeah.
Okay. So, and then now they do this in the bitter neurons. They measure activity of brainstem bitter neurons. So SST, Cree, GCamp, vibrophotometry, activate with channel rhodopsin in the bitter cortex, and they see slight potentiation of the bitter signal in the NTS. Right. In figure E, they go to the central amygdala, which in figure 1 was shown as projecting to the NTS to see if this could suppress sweet neurons. So here what they do is they're injecting channel rhodopsin in the bitter cortex and then shining light in the central amygdala. So they're stimulating the projections of the bitter cortex neurons and then measuring activity in the sweet neurons in the NTS. Right? And even just by doing that, they can suppress the activity of brainstem neurons to sweet.
I have a question. Since C and E basically have the same results using the same experiment.
C and E, yeah.
Yeah, the GCBT neurons and the CEA neurons basically are doing the same thing. Why is there two? Does this GCBT project to the CEA, which projects to the NTS, or is it like either or? Like, how does that work?
Probably both, right? Because even in the diagrams, they show that there's the GCBT that projects directly into the NTS, and there's another parallel pathway that goes through the CEA. And here they're doing this with the other pathway.
Do we know?
So
like
in C, it'll be affecting both pathways if there were such thing as both pathways.
Okay, but why wouldn't, if it's faster for it to just go straight to the NTS, then what's the point of it going through the CEA as well?
I think it's because generally people think that the central amygdala is used for aversion, and that this fits in nicely to how bitter might cause some kind of aversion to suppress activity.
Do they really mention the CEA central amygdala activity upon the stimulation of gustatory cortex?
The activity in the CEA?
Yeah.
Is
there suppression or excitation from insular or whatever?
Oh yeah, but that that was shown in a different paper. Right? The Li Wang et al. Paper shows that there's a bitter cortex to CEA projection.
And that is excitatory projection.
Yeah, yeah.
Okay.
Okay. So what happens in figure 6?
Electrophysiology.
Go ahead.
Go ahead.
Yeah, good.
Oh, they are doing electrophysiology to test if these neurons are actually directly inhibited or activated by stimulating the neurons in the central amygdala or GCBT region. So idea here, I think, is they are injecting a Cre-dependent TD tomato so they can see the SST, the beta neurons or the COB2 sweet neurons in the section. I think they did it in the sections. And
like slice recording.
yeah, slice recording. So what they do is in panel B, that's the schematic is that from the the upstream regions that are projecting directly down to the down to the rostral NTS neurons, they are just using electrode to measure the activity. So in C, we can see that when they use, since there are channel dopsin expressed, when they are shining light on upstream regions, they can see that there is an action potential. I think it's an excitatory, right? EPSCs in the beta neurons. So in C, when you activate the GCBT neurons, you get action potential. However, if you use a drug that blocks the AMPA receptor, then this this current is just disappeared. So, quantitative study in panel E, it just proves that these neurons, SST, TD tomato neurons, are regulated by those thing for these sweet neurons, and they find that it is an inhibitory current. So it's an iPSC in G. That means that there will be an inhibitory regulation from the central amygdala on the sweet rather than excitatory in the bitter. And they find that if they provide the same AMPA receptor blocker, the DNQX in panel H, it doesn't even at all change the current. However, if they provide a GABA receptor and antagonist PTX, then they see the current disappear. So it proves an inhibitory current. So that further proves their hypothesis that the sweet neurons in the rostral NTS is inhibited by the central amygdala, which proves their top-down regulatory model. Yeah.
Thank you, and we'll move on to figure 7. Jaha, you want to have a 2-minute go at it? I have to go into a seminar.
Okay, so
yeah.
So now they come to a point that CA also important for the sweet responses in our rostral NTS. MBQX, as shown in previous figure, block the excitatory transmissions. It basically also means any other excitatory drive to the CA. So that's basically silencing the CA activity. In B, they see the effects about applying the MBQX, and this basically, wait, I had to say about it. So ACK sweet 100%, and then before applying the MBQX, I don't know what.
This is with sweet and bitter, isn't it?
Yeah, it should be a mix.
Yeah,
it should be a mix.
Yes, yeah.
So even pre, it should also be the mix, right?
Yeah.
Okay, then that makes sense then. So pre-mix and the effects also. So mix means basically means the ACK plus the quinine, and then inside the mix bar, it means also get some side staining, and MBQX is added another time. So MBQX basically blocks the suppression of quinine.
Yeah.
And then C shows us in a similar way applying the MBQX in the CA and see how it affects the leaking behaviors of the mice corresponding to either the pure ACK or quinine or the mixture. And what they find with the MBQX in the middle of the D is actually in the mixture is actually increased, like they restore the leaking to the level of the ACK itself, so comparable. While if the MBQX, if this effect somehow is washed out, after the post infusion, the mixture, the quinine effects, the suppression of quinine intake on the leaking behavior is come back. So basically means the first panel says the I think it's the electrophysiology. I'm not sure. And then C is the C, and this row is for behavior, like is the consistent. And then E shows us what about using another way to inhibit the CA. So they use the more maybe temporary controlled ways using the night, and they infect the CA neurons with two different version of halorhodopsin, which is you can take it as the reverse, like the antagonist of the channelrhodopsin. So channelrhodopsin excited neuron, halorhodopsin inhibits neurons activity. And with this, they shine light on the terminal of the ROS20S instead of CA itself, because CA can send projections other than in the ROS20S. So they have a local light illumination, so they have local inhibition onto the ROS20S. From F, you pretty much see a similar, let's say, restorement of leaking behavior upon the light shining with the orange bar. So orange bar restore its similar level as the ACK at the right side. And their quantification on this mixture, they should have like a show us the significant, whether it's a statistically significant increase. Presumably it should be so. And if G is a control for those halorhodopsin, it means like there's no change for the leaking behavior upon shining light with the GFP control construct. That's it.
So overall, the message is pretty simple. They found two populations in the NTS, the bitter marked by SST, sweet marked by CALB2, and that these cells get modulatory regulation from the cortical and the amygdala neurons. Right. Does anyone have other questions or comments about the paper?
If the gustatory cortex mainly excites the SST neurons, right? While CA mainly inhibits CAB2 neurons. Is there a connection between those two neurons, which upon because the author shows activation of gustatory synaptic input actually inhibits the CAB2 responses, right? I'm not sure they're doing the local activation or like a global activation of the gustatory cortex. If local, then that means there is local interactions between the sweet and bitter populations in the ROS20S itself.
Wait, how do you get to the local interaction in the NTS?
Oh, can you go back to previous figure showing that activation of gustatory cortex reduce the sweet responses in the NTS? Yeah, like this.
Yeah.
So the light stimuli where's the figure for? Okay, it's global. Okay, then that makes sense. It's global.
You mean this one, the C?
Yeah, C. Yeah, it is global. If it is, for example, if they put the CHR2 shining light at the ROS20S, while recording the sweet neurons.
Mhm.
Based on their
Oh, I
model,
see. Yeah, yeah,
they could
Yeah.
do this.
yeah.
Okay.
Yep.
And also in their model also shows the projection from gustatory cortex, I think is unilateral, ipsilateral.
Mhm.
CEA is contralateral, right?
Mhm.
Is that right?
I think so.
So I think I need to oh sorry,
It's the reverse.
it's the opposite.
Yeah, yeah, it's the reverse.
CEA is ipsilateral,
Yeah.
gustatory cortex is contralateral.
Mhm.
Okay.
Right.
But it is the same contralateral insular cortex target the CEA.
The contralateral.
So what about the
Oh, I see.
insular cortex?
from the gustatory cortex to the CEA.
Yeah, is this contralateral or ipsilateral?
That must be IPSI, right?
Okay. I'll just check this out.
Yeah.
Yeah, that must be ipsi.
Yeah, that I would imagine that's IPSI. Okay. Okay, good. Next Friday we'll have the last journal club of the year. So stop sharing this. And I'll see most of you in the seminar that starts right now, or started 2 minutes ago. Okay. All right. Thank you, everyone. Bye.
Yeah.